Okay, so during detoxification, which we'll look at in a little bit more detail as we go along, we go through two pathways called phase 1 and phase 2. So phase 1 is a hydroxylation pathway by adding an OH to a toxic substance, so it makes it more water soluble, and then we conjugate it, usually with a protein like glutathione and amino acid, or sulfur, some substance like that, glycine and acetyl group. But it's this first family of detoxifying enzymes called Cytochrome P450, which we're particularly interested in. And these are the ones that metabolize most of the drugs and chemicals, strange chemicals within our body called xenobiotics. So drugs have to mostly go through these pathways, as do other chemicals. And we know that green people are very, very sensitive to chemicals, to drugs. And I couldn't understand why green people particularly should have been born 200 years ago when the world was a much cleaner place. And also why in Scandinavia, where practically everybody's weakens to the green, they're all tall, but it's incredibly clean. And that's why, because pollution, they can't handle the pollution very well. And then I discovered that when we got the different major detoxifying enzymes to test with, was that green people had positive on cytochrome P450-3A4, which is the most common one for detoxifying a lot of medication and poisons, and that 50% of drugs go through this particular pathway, and that green people strengthen to that particular one. And I thought, I wonder if genetically we have different expressions of cytochrome P450 to the different constitutions. So by working through people who've got chemical sensitivities, we found that cytochrome P450 2D6 was also deficient in green people, and to some extent cytochrome 2C9. And this added up to an awful lot of people. 80–90% of drugs were not being metabolized very well for green people. So we did the same thing for red people and for blue. And I found that blue people strengthened to cytochrome P450 1A1 and 1B1. Okay, now 1B1, as we'll see shortly, is very important because it metabolizes estrone and estradiol, the two active estrogens, into the hydroxylated derivatives. But more important, it drives it into the 4-hydroxylation pathway, which is the pro-malignant one. Okay, so here we've got a situation where people who are blue, and that's why I color-coded this chart is available on the website, by the way, to download. All you've got to do is download it. It'll come out as an A4, but you can then blow it up to an A3 and have it laminated. And it's quite useful. It works 100% with people. If they've got a sensitivity, you just put on the different cytochrome P450s, and one day we'll be able to actually match that to all the drugs in the British Pharmacopoeia. And you'll be able to say, this is a green person, and this person potentially could have a problem with this particular medication, and it may not be the best of ones. Luckily, we found things which will help reduce and stimulate these pathways, and very often spices are very good here. Spices seem to upregulate the cytochrome P450 pathways. And we'll see a bit later on which spices are good for blue people, because that's what we're talking about, but there are spices which stimulate the cytochrome P450s in red and in green people as well. Okay? So this is something we found that blue people with that biotype have problems. They have the short straw, if you like, of the 1A1 and the 1B1. That does not mean they don't have the enzyme. Okay? If we imagine, because we've got 3 constitutions, we could have 3 expressions potentially of that gene. Maybe there's more expressions of it in some genes, and therefore the enzymes associated with those genes may not work as well. Okay? So some of them will work a little bit. We know this with ApoE4, the lipoprotein to break down cholesterol and metal fragments and things. ApoE4 is the poor expression, so we get high cholesterol and things with red people, whereas ApoE2 and ApoE3 are better expressions. And it's the same here. It doesn't mean that a blue person doesn't have 1B1 or 1A1. It just is not a very good expression. We'll call it the short straw, if you like. And we'll see more importantly, or in addition to this, there are many enzymes which blue people have the short straw on. Okay? And there's many enzymes that they have the big straw. Okay, so there's good and there's bad in every. Because people say, well, what's the best colour then, Chris? What colour should I be? And the answer is no colour. You shouldn't weaken to any colour, because if you're at that elevated position of love, completely up there, unconditional love, where David Hawkins put people at above 540 on his scale, you wouldn't feel unloved, and you wouldn't feel not loving, or you wouldn't feel not lovable. You love, and that's it, isn't it? That's unconditional love. And the few people who are there are perfectly balanced in their right and left brain because there's no conflict, because there's only unconditional love. And they don't weaken to a colour. Okay? But you won't find many of them. And there's none in the government. Right, now, general dietary rules. And we're just going to go into this because then you'll see how this relates to cancer as we go along. So, generally, we found on blue people, they're better for low protein because they have low hydrochloric acid, therefore they have low pepsin activation. Plenty of fruit and veg, yeah. No cow's milk or lactose. Lactose is the key thing here, not so much casein, so they can have the protein, and as we see, the importance of things like cottage cheese are fine because they're low fat and they're low lactose. So you can usually manage that. Beware of old and aging cheese. So it's the old ones, the mature ones, which are not very good here. Avocados, bananas, and chocolate. That's because of the tyramine. So this is probably less important with malignancy, but certainly tyramine foods. They're not very good on aspartame. So aspartame is the artificial sweetener, and monosodium glutamate, which is the synthetic salty substance. So they have, again, because of the enzymes deficient in detoxifying, they don't seem to detoxify these very well, and so they're the ones that get hyper. Okay, they can't break the aspartame and the MSG down. And we talked about limiting alcohol. Now, back in the summer, I was playing around with these different red, green, and blue types and seeing if there was anything which was a theme which could stimulate the cytochrome P450s. And I went through, first of all, I think I did it with the oils, and because they were consistently different fatty acids for a blue person, a red person, and a green person, I tried it with the vitamins and the minerals. And what you find is that when you go through the product kit, that there are certain things which are common to red, green, and blue people. And all these people, all blues, okay, here, we're not talking about malignant, we're just talking about the blue body type here, strengthen against the blue acetate to thiamine or B1. Okay, and I thought this is really interesting. I didn't have a clue why, because I know B1 works in the energy pathway, works in other pathways as well, but primarily in that conversion of pyruvate to acetyl-CoA. But now, of course, we know that is the hypoxic area. This is where the big problem is with all malignancies, is they have a build-up of pyruvate and a decrease in acetyl-CoA, which shunts it, as we'll see in detail later on, into lactic acid. Okay? So of course these people need B1. Okay? And when you look up in the books, whole grain cereals, liver, yeast, who eats liver? Who eats yeast products? Who eats whole grain cereals? Very few people in the population, no. Okay? So it's a miracle, again, most people get enough B1. Many, many people are hideously deficient in B1. And we'll see the role later on of B1 in the immune system. So in other words, if you can't make in your cells enough to spin the Krebs cycle, okay, how the hell are your immune system cells going to work? They've got no energy. You know, the rest of you's got chronic fatigue, well, your immune system has as well. Okay, it can't produce the oxidative derivatives that it needs to do the killing. Folic acid, we mentioned about folic acid and genetic expression or healing and making new cells. B12, I mentioned that, B12, why B12? Because it's hypoxia, okay? It matures the red cells. So the standard B12 or hydroxycobalamin is the base substance that the body needs. Most cases it can activate it into the coenzyme forms itself, but sometimes there's a polymorphism and they can't. But B12 always shows up, as does choline. And we'll see why choline in a minute, and because the major regulator of the immune system is acetylcholine, or the one that regulates the parasympathetic system, and we make acetylcholine from acetyl-CoA. And we'll see that we make acetyl-CoA from pyruvate. So when we convert pyruvate into acetyl-CoA and then on to making acetylcholine, we need choline. Okay, if we don't have choline, we can't make acetylcholine. Okay, so blue people, very, very important. 4, very key. Those are the only vitamins that they show to consistently. Now, they may need a little bit of this and a little bit of that in addition, but every blue person that you test will show to those 4 vitamins. Now, minerals. Interestingly, boron shows them more. Now, we know nothing about boron, In Europe, it's not considered an essential nutrient. In America, I think it is now. You know, very small amounts, 1 milligram, 1.5 milligram. It's known to help estrogen and metabolize estrogen, particularly in low estrogen postmenopausally, and has a role in bone density. That's all that's written about it. But probably boron has other factors as well. We know it's involved with hair greying. Okay, it's one of the most important factors, deficiency when hair starts to go grey. But we don't know how it works, but we know that boron deficient animals go grey. Okay, iron. Now this is the only colour constitution that needs extra iron. Why do we need extra iron? Probably to make sure the hemoglobin levels is okay. Okay, to get that hemoglobin, to get that oxygen to the tissue. Magnesium. Magnesium works in over 75% of all enzyme pathways as a cofactor, but certainly they need a lot of magnesium. Okay, it's again the only colour that consistently shows magnesium. Magnesium is involved with energy production. You know, most of the pathways of energy need magnesium, magnesium, magnesium. Selenium. Selenium is a great detoxifier. It's the cofactor in glutathione peroxidase, which helps us to be able to conjugate toxins, particularly hormonal toxins. And selenium was also a great linker to certain toxic metals to get them out of the system. We know it converts T4 to T3 also in the thyroid. Sulfur, one of the major detoxifiers. Blue people need a lot of sulfur, okay, and therefore they need a lot of sulfurous foods. And one of the great things with the cancer is the Budwig diet, which is cottage cheese. And cottage cheese is loaded with sulfur because it's very high in cysteine, the amino acid which has got lots of stuff. So sulfur is a really important detoxifier for blue people. And zinc, which shows up for all three colours. Everybody needs zinc. You know? So a little later on we'll do the zinc taste solution test on you, and I'll guarantee 75%, if not more, will not taste zinc, particularly this time of the year, because we haven't had enough zinc foods in from the soil any fresh. And zinc is needed primarily in the body for the repair of tissue through that enzyme called DNA polymerase. So when your DNA is oxidized by free radicals, to repair that DNA, you need DNA polymerase, which is a zinc-dependent enzyme. But blue people need it also for the opposite, for the stimulation of the cell apoptosis. So without apoptosis, we can't destroy the bad cells, and we can't repair the good ones. So zinc levels are really, really critical in this case. Now, what we found, probably originally when I started doing this, was very interesting with the different oils. We know that blue people and fish oil don't go together, okay, simply because of the mercury. It's not the fish oil, it's the mercury within the fish oil which is the problem, okay? So we looked for seed oils or non-marine sources of oil. And we find that the three that show up is flaxseed. Now flax is 60% alpha-linolenic acid, which is an omega-3 fatty acid. Pumpkin seed, which is a 10 to 15% omega-3, the rest is omega-6, and walnut oil, which again is 10 to 15% omega-3 alpha-linolenic acid. So these are all omega-3 fatty acids, non-marine. And what you find with the blue person is if you've got the old fatty acid kit, is you will find that they will strengthen all blue people to alpha-linolenic acid. Now most people will convert alpha-linolenic acid into the longer chain derivatives, as they're called, of EPA and DHA. Okay? Now young people have difficulty here, up to the age of 7, of getting these enzymes to work. The delta-6 desaturase and the elongase enzymes need enough cofactors, magnesiums and zincs and B6s, to get them to work. But the vast majority can convert alpha-lipoic acid into EPA and DHA. And more recent research has shown that the amount of EPA and DHA is much, much less than what they thought. The need for converting is much, much less than what they thought. Drowning yourself in EPA and DHA was not a good thing. It actually caused imbalances in the phospholipid membranes. Okay, so this idea of having grams and grams and grams of fish oil is now being dismissed as probably not very good for the health, and in fact may actually block the oxygenation, as we'll see later, of the cell membranes to getting the oxygen out of the red cells into the tissue. What is very important is to get the balance, and this is the great question, is the balance between the omega-3s and the omega-6s. So too much of one, too much of the other may be a problem, but even more so, and this is what we'll be emphasizing later on, is when you alter these oils and you twist the molecules. So we know that an unsaturated has a kink in it, and when you heat that unsaturated, the molecule remains the same, but it twists, and then it straightens out. And when a molecule, an unsaturated, straightens out, it acts like a saturate. Okay? So it has a higher melting point. In other words, it makes the membrane stiffer. Okay? So if you heat a vegetable oil, you'll convert it into a trans fat, which will have a higher melting point. And it means that the membrane will be stiffer, in which case oxygen can't get across. Okay? Imagine the doors there, those two fire doors, as the way the oxygen is going to get out of the red blood cell. It has to go through like a plasma. You know that magic screen in the Matrix where people walk through and they come out the other side? That's what oxygen has to do in the cell membrane. But the door is shut because it's stiff. Okay? And if it can't get out, the cells are starving, even though the red cells have got the oxygen. So on the oximeter, the red cells are okay, but they're showing all the signs inside of a lack of oxygen because they're not getting across the cell membrane. And what it is, it's the balance. And the balance is different in me than it is in you. Okay? So we all have a different oils that we need, but they fall into the different constitution. The DNA variation in humans is only 10%. The difference between you and I is 10% max. Okay, it's not very much. What is that difference is the expression of the genes. In other words, have you got the short straw and I got the long straw on certain things, or have you got the long straw and I got the short straw? That's the variation. In other words, which oils do you need and which oils do I need, which vitamins and so on. And the answer to blue people is they need the omega-3s in greater amounts. Proportion here, by giving equal proportions here, these oils, this is 60%, this is 10, let's say that's 10. Add them all together, it's a 4-to-1 mix. There's virtually no omega-9s or oleic in those oils. So a blue person is heavily loaded more towards the omega-3 oils. That's what they need. Okay? Now, when you come to looking at a green person, which I am, we need the omega-6s. We need the sesame, the peanuts, these are all written down, hazelnut and grapeseed. When we test our, it's linoleic acid we need. We don't want alpha-linolenic acid. Flax does us no good. In fact, we rebel against flax. Now, the red person is different again. They need some omega-9s. They need principally omega-6s and omega-9s. So they need the flax, a little bit of flax. They need pumpkin, and they need olive oil and hemp. Okay? They're the only ones, the reds, that need olive oil. They love olive oil. That's why they all live in the Mediterranean. Okay? It does them a great deal of good. This is what the Italian diet, the Mediterranean diet's about, is olive oil, isn't it? It's omega-9. There's no omega-6s and no omega-3s in it. But they love it. So, very, very important with the red person. They can use olive oil. It's one of the best oils. But they must not cook in it. Because if you cook in it, what do you do to the oil? You change it into a trans from a cis, and they'll harden up. Okay? So what do the vast majority of people in this country cook with when you ask them? Olive oil. Okay? So they mustn't cook. So the blue person can cook in olive oil. No problem to a blue person to cook in olive oil, because it doesn't matter if it goes trans. They don't take it up. They don't need it. Okay? What they need is the omega-3s. Now, you're not likely to cook in flaxseed, pumpkin or walnut anyway. Okay? But as a blue person, when did you last eat any of those oils? You know, the dry weight of the human body is 30% fat. 30% of the dry weight. When did you have any oils? Oh, I have a salad dressing. Did you? What did you put in it? Oh, olive oil. I'll answer it later. Right? Okay, olive oil. That's no good to a person here. Okay, it's no good to a green person, and it's no good to a blue person. But it's fine for cooking. Okay? So what should the red person use for cooking? Not olive oil, obviously. Sunflower's fine. Anything, apart from the ones that they need. Okay? So you mustn't cook with the ones that you need, because your body won't know the difference between the trans and the cis. It takes up on the phospho part into the cell membrane, not the fatty acid. It doesn't know the difference between a trans and a cis chain. It knows the difference between a phosphatidylcholine and phosphatidylinosytol and so on, and it locks it up. So if you have a diet rich in Dunkin' Donuts and things, you will take up Dunkin' Donuts into your phospholipid structure in your brain and other tissues. Right? So very, very important. Cook with the oil that you don't need. Okay, so for these people, they can cook with olive, etc., but they need spoonfuls of this. And when we come to testing it, it's not a teaspoon. I was amazed. It's not a teaspoon. Most people need between 4 and 6 teaspoons, okay, of their oil in their diet every day. Okay. Now, when should you take an oil? when do you produce most lipase in the body? Is for supper time, for dinner time, between 7 and 9. Okay, you have oil for breakfast, it'll just, ugh, you know, they'll heave and that. Even at lunchtime. If you have your oil in the evening is when you have the maximum production of lipase to chop those oils up into their fatty acids so you can get those essential fatty acids into your system. Okay? Don't take them out at that time. Children is a little earlier. Children will probably be in kidney time. For the rest of us, it's circulation sex time between 7 and 9 in the evening. Okay? So oils are really, really critical. And they're more important, as I say, because you'll need to oil people with malignancies up. And why do we need to oil? So the cell membranes can work. So they can get the toxins out of the cell membranes and get the oxygen and the nutrients in. So you find this is really, really critical. And probably one of the most critical things that you can do for people is to put them on the right types of oils and the right proportions. Now, the idea of 2 to 1, you know, we've even heard 1 to 1, 1 omega-3, 1 to omega-6 banded around. That would just be so toxic for people. Okay? They say, oh, this is what Eskimos do. Eskimos don't get heart attacks. Right. If you've ever been up to the Arctic Circle and seen an Eskimo, they look 40 years older than what they are. Okay, and it's not just the cold weather. They only eat seal blubber and things like that. They're getting omega-3, omega-3, omega-3. And that's good because it keeps them warm, because it keeps the blood thinner. But they get no omega-6s hardly, which keeps our skin nice, so their skin ages. Okay? So for us, for the blue people, okay, these need these oils and only those oils. Herbs and spices, again, very interesting. Basil, lovely herb. Coriander, a great detoxifier. Cilantro, the Americans call it. Great chelator of toxic metals. Very important to the blue person, as we'll see. Not just mercury, but there's that little bit of nickel, but most important is the arsenic. Getting that out. Chemicals, they need the hot ones, the spicy ones, in order to upregulate those cytochrome P450s. They need the chilies. Okay? Looking more towards the Eastern diet, isn't it? Where do most of our spices come from? India, China. What type of people constitution are Indians and Chinese? They're blue. They have to upregulate their cytochrome P450. Chili, cinnamon, and paprika. Lovely spices. So these would be the herbs and the spices you favour culinary-wise, but also as a tea. So by making a blue mix of these, it can be taken as a beverage. Now for the majority of people, again, it needs to be a reasonable amount. You take it as a tea in hot water, and you put it in and you have it 2 or 3 times a day. But it's quite a lot. It's 50, 60, 70 drops. It's not a couple of drops in there. You need to not only flavour it, but you need it to actually work. So to get it to work, and you do the dose, you know, it's about a teaspoon of the actual liquids, which is a blend of those 6 different spices and herbs. Now, the red people and the blue people need different ones, okay? Because their cytochrome P450s and their toxicity side and their detox side are different, okay? So they need different herbs and spices. But today we want to talk about the blue person. Probiotics, again, very important. We now do a range of 6. So we've included Acidophilus now in our range, Rhamnosus in our range, and Bifidus in our... we've got Bulgaricus, that's it, Bulgaricus in the range, and we've got Bifidus, Bifidobacteria. But the blue people tend to favour the Casei, which is the Kaffir one from the Caucasus Mountains. So again, it's one of those probiotics which is more into the sort of Mediterranean type of regime. And they make a curdled type of milk called Kaffir, and it's supposed to make you live to 120. Right, so that's the blue constitution. So I have not yet found a person with a malignancy who is not completely blue or blue in one eye. Okay? Now we talked and we saw here a hybrid. Okay, so the hybrid was red and blue. Now sometimes Jill has a tall, thin son who's 17, and he was blue-red, wasn't it, or red-blue? Red. He was red-blue, okay? But he's 6'4" and thin, okay? But he was manifesting signs of being the red was his dominant. Why was he thin? Because he was thyroid, okay? The red goes with the thyroid gland, but he was tall. So what should he be? At 6'4", he should be green, okay? So when he was zapped, he became a green, okay? So he was never a blue or a red. So some of us are not who we think we are, okay? What does it? What changes a person's colour? I showed you earlier, okay? Their thinking process, okay? So when you've been under a lot of stress And kids go through a lot of stress at school, like examinations, all sorts of things, growing into a teenager, all these things are big, big stresses. Okay? Now, your other son was green, blue. Red, green, okay? Again, 6'2", 6'3", but totally different build, Mr. Muscles himself, okay? So he's a green, he's typically of the green. But he had the red aspects are there, again, with the thinner part, the taller bit, but the red aspects receive much more of the thyroid. Not an ounce of fat on him, okay, but all pure muscle. So hopefully there's no anabolic steroids in there somewhere. But you see, we can change. You get tall people who are thin, tall people who are broader and muscular. But tall people should be green. Medium people should be red. And the shorter people are the blues. So think about the blues coming more from the sort of Far Eastern body build. Think about the medium height ones like the Mediterranean, Celtic, Caucasians. And the greens, more the Scandinavian, Viking, raiders and pillagers who come in and blended with the British in 900. Okay, now let's change aspects now. We looked at the constitution, which is what we physically see with the person in their height and their bone structure. Now let's look at the things that can actually affect the gene itself. So the gene mutation, this is what we're looking here. What makes a gene mutate? Okay, so we've looked at the type of body build of a person. We've looked at the reasons behind that and the emotions which make us into who we are of our expression of love. Now we want to know is what actually happens at the gene level. And most of you are going to be familiar with a lot of this work because it's very easy using kinesiology or EAV and things to determine by using markers whether a person has a polymorphism or a mutation in the gene. Now, the first one that I came across and I think was worked out was this particular one here in the folate cycle. The first SNP to be involved with a malignancy was a U into a T, it's called. Now, this is folic acid, which is not, it's a vitamin, but it's not an essential, well, it is essential, but it's not an active vitamin. Because the body doesn't use folic acid on its own, it has to be activated into its coenzyme form. And its coenzyme forms are tetrahydrofolate, methylene tetrahydrofolate, and methyl tetrahydrofolate. Okay, and here we've got methanol tetrahydrofolate or folinic acid. So methanol is CH. You might want to just jot this down because everybody gets confused with it. So methanol or folinic acid is CH tetrahydrofolate. Methylene is CH2 tetrahydrofolate, and methyl is CH3. Now we need methyl here to put a methyl donor onto vitamin B12 to recycle homocysteine, and we'll look at that with the methylation pathway. So methyl groups are very important. So how do we convert folic acid into the active forms is we add hydrogens onto them by an enzyme which is B3 dependent here and needs a bit of vitamin C to add 4 hydrogens to folic acid called tetra or 4-hydrofolate. Now to add the first CH2 onto that we need a methyl donor. And this methyl donor comes from the amino acid serine. And the serine changes to glycine and releases the CH3 en route. And this enzyme needs B6 in order to be able to do it. So here we see a B6 connection in activating folic acid. And to activate B6, we know we need magnesium. So a person who's magnesium deficient could have a deficiency of pyridoxal 5-phosphate, which doesn't allow folic acid to mature or to activate, and that then won't recycle B12, recycle homocysteine. So you could have a whole chain. So where is the problem with the person? So luckily, using muscle testing and things, you can find, the problem is a deficiency of magnesium, or there's a polymorphism and they can't activate the folate, or they can't activate the B12. So luckily, from a functional point of view, we can determine this. Now, if you don't get enough methylene tetrahydrofolate, you haven't got the methyl donor to put onto uracil, which is a base substance in RNA, to turn it into thymidine, which is a DNA. Okay, so in other words, you get a little bit, the uracil component of RNA getting into the DNA. And that certainly won't work, will it? In other words, it will read as a blank. You can't actually activate, the genetic sentence can't be read. So this thymidine synthase enzyme requires the methyl group from methylene tetrahydrofolate. So what you get is a U into a T position, okay, or polymorphism or SNP. And that's why it's called U into T. And this was the very first one, and this is the one that got me thinking, well, I can test that using the bases of the DNA and RNA to see what the polymorphism and how many people got it. And this was the first one with breast cancer. So in cases of breast cancer, more often than not, you find a U into T polymorphism, which encodes that they need a greater amount of folic acid, but more specifically, the active form, which is methylene in this case, tetrahydrofolate. So it was this pathway which drew our attention, because this was blue people, and if blue people are developing a malignancy, you know, like a breast cancer and things, this pathway is very important. How do they activate the folate? So this little bit really drew my attention. So what we found in the books, that serine, the amino acid, could be substituted by hydroxymethylarginine, which is rich in grapes. Now, later on, we'll talk, there is a grape diet, and this may be one of the reasons. And cruciferous vegetables, that's your broccoli, your cabbage, your cauliflower, and your Brussels sprouts, okay? And beetroot, very important. There's a lot of schools of treating cancers using beetroot. The good news is you have to have beetroot 3 times a day, in usually all orifices, in there. The bad news is it's got to be raw, okay? So it's usually raw, and they don't recommend just the juice. But there are clinics which claim great results in Russia, particularly, where they love the beetroot, the borscht soup and things, and they use beetroot. But bring beetroot back into the diet is very good, particularly if you're a blue person. But it must be organic. You know, I've eaten beetroot and end up with great rashes from whatever they use as the spray or the pesticide on it. You must get organic beetroot, don't get non-organic, because all root crops absorb the toxins, the pesticide residues from the soil. So if you're using root crops particularly, make sure they're organic. But we can supplement here our grapes, our cruciferous vegetables, and our beetroots as a source instead of serine. So this may be one of the importance of bringing this into the diet as a preventer, you know, particularly for blue people. So these are great inputs. Okay, so single point mutations are usually when the nucleotide rather, has a different base in it. And if it's a single one, it's called a single nucleotide polymorphism, or a SNP for short. Or we can get deletions, insertions, and rearrangements where groups of these are put in the wrong place. I call these like word programs, cut and pastes, but it's the single point ones. So they could be a single transition where adenine goes into guanine or guanine into adenine, or cytosine goes into thymine or thymine goes into cytosine. So they're just reversals of the position that they should be in. Or you could get uracil from RNA going into thymine, which is what we just looked at there. Or we can get transversions where you get adenine going into cytosine, guanine going into thymine. So we can get a pyrimidine going into a purine or vice versa. So single base changes or mutations may have no effect at all. If it's not on the active reading part of the gene, it may not be detected and you may not notice any change. Or there could be just a little bit of nonsense, or there could be complete nonsense and the gene can't be expressed. And therefore the enzyme that it encodes for can't be read at all. So we could have a polymorphism with it partly being read, and also we could have it where it has a serious effect. Luckily, if it has a serious effect, most times this is if it's inherited, these children don't survive. Okay? What most people have is a missense one where you like drawing the short straw, so you've got a little bit of a mistake in it, and therefore you can't express the gene correctly, and therefore you don't produce the right amount of protein or enzymes. So what you would do from your challenge point of view is you would use your base substances. There's no need to use the whole nucleotide because it's the base which you're after. You challenge each of the 5 nucleotide bases from strength to see which one weakens. So what you do is the one that weakens is the bad one. Okay? Now what we do is note which one weakens. Adenine, cytosine, guanine, thymine, and uracil. So you put them on singly. Right? So if it weakens, it means it's something detrimental. Okay? So that would be the bad guy. Then you leave the bad guy on, and you put the other 4 ones one at a time on, and find out which one strengthens, okay, or negates that weakness. And that gives you your combination. It's as easy as that. So challenge weakening nucleotide base against each of the other nucleotide bases and find which one negates. So for instance here, G weakens, guanosine weakens, and is negated by T or thymine. So this would be a G into a T. This is one of the most common polymorphisms around and encodes for a greater need for pyridoxal 5-phosphate in the system. So in other words, this person needs a greater amount of the active form of B6 than what they can biologically make. So there are times in their life, particularly when they're under stress, that they may need to take more P5P or pyridoxal 5-phosphate than what they can just get from their brown rice and their vegetables from their diet. Okay? So this is the person. So everybody with a mutation has a coenzyme necessity of a greater amount of it than what they can probably get from their food. So in other words, everybody with a polymorphism or a mutation needs a little extra. Okay? So that extra very often is the active form of the vitamin, like pyridoxal 5-phosphate or adenosylcobalamin, etc., rather than just the normal vitamin. So there's always an associated coenzyme for each one. So each SNP defect may be apparent in nutritional deficiency. So what causes the polymorphism in the first place? What causes the mismatch is we either inherit it, what in homeopathy they call a miasm, in other words, an inherited defect from mother or father's line, or we acquire it. So we may acquire a polymorphism or a SNP due to zinc deficiency because we can't activate DNA polymerase. Okay, so we're zinc deficient. Very, very important, this zinc deficiency. Okay, to repair the DNA which is damaged by reactive oxygen species. Or we can be exposed to certain toxins, and these toxic reactions create the generation of free radicals. And the main ones here are from the pathogens will be viruses, what we talked about, because they are strings of DNA or RNA, toxic metals, and today we want to particularly think about mercury and arsenic, mycotoxins, particularly aflatoxin, which is highly carcinogenic, which is the mold from fusarium, which is often on nuts and grains and things, okay, which we take quite a lot of in our diet all the time. It's very, very carcinogenic. Chemicals or strange chemicals, xenobiotics in the diet, and ionizing radiation. Okay, so these are the main enemies to the gene. These create the reactive oxygen species or free radical reactions which damage the gene. Okay, that damage then has to be cut out and a new base put in. Okay, it's as simple as that. It's like repairing the side of a ladder. Now, if there's damage to the side of the ladder, what is called a nick in the side of the ladder, you take that damaged bit out and you put another piece of wood in. But that piece of wood has to fit exactly. Okay? Now, if you don't have the right amount of zinc and folic acid, you may slap any old piece of wood in there, and in other words, another piece of wood that doesn't fit properly, but it fits in there. And then the ladder's lost its structure. Now, some free radicals do damage on one side of the ladder, like superoxide, but other ones damage both sides of the ladder, and then the ladder collapses. And that's the dreaded hydroxyl radical, and peroxynitrate can damage both sides of the ladder and do a lot more damage than peroxides and just superoxide. So the reason for the reactive oxygen species or free radicals is the toxins or the chemicals and the viruses, etc. So to repair, there's only one thing we need to think about is zinc, zinc, zinc. Now 75% of the room are deficient in zinc. When we do the zinc taste, it's a good time to think about doing this. Jane, if you've got some zinc taste solution here, we should have it. No, we have. Zinc taste. Yeah, go and ask Clive. Not the zinc, but the zinc taste solution. So the zinc taste solution is a diluted 0.1% solution of zinc sulfate, and is a traditional Bryce-Smith method where you squirt some zinc onto the tongue and you leave it there for about 10 seconds and then swallow. If you get a yuck taste, then you're okay in zinc. If you get a mild taste or no taste at all, then you're zinc deficient. Okay? But even with the taste and it's yuck, you can't say 100% the person hasn't got zinc deficiency. All you know is that they're okay for zinc in the tongue. But it doesn't mean in the rear part of the brain or the big toe, they're all right. So don't base it, muscle test the person or EAV them to see if they strengthen to zinc. That's your better test. If a person strengthens to zinc, and my experience is red people, blue people, green people, all strengthen to zinc. Because how do we get zinc? The only source of zinc these days is the sea. Okay, all the whole grain cereals, fruits and things, eggs that you talk about, you know, when do they put these zinc back in the soil? It's gradually getting more and more depleted. So the best source of minerals now is always the sea. So seaweed extracts oysters. Oysters are a good source. Seafood of any type is a good source of zinc. Okay, nuts are a reasonable good source of zinc. Okay, so here is the genetic code. So we see the different amino acids here, and what they do is every genetic sentence has a start, and the start to every sentence is the code for methionine. So methionine is the first start of every sentence, and then we have a full stop. And the full stop will be one of these codes, okay, at the end there. So let's just take an example of a polymorphism. So let's say we had this one, CTC. Okay, so let's have CTC here. No, let's choose another one here, one which is less common. You see these ones which have several codes. These are the big amino acids in the body. So leucine, you can see, has lots of codes, and leucine is the most abundant amino acid, followed by isoleucine, followed by valine. These are our branch chain amino acids that give us our muscle bulk, etc. So if we take a smaller one, like proline, okay, here, and let's put in proline here, and let's put a polymorphism into the C position here. So instead of CAT, I'm trying to find another AT there somewhere. Oh, okay, so say we have here, where were we, CAT. So say instead of the C in there, we have a polymorphism, and we get a G into that position instead. Okay, so we've had a polymorphism, we slipped a G into the C position. So instead of that encoding for the amino acid of proline to make amino acid structures, including enzymes, we end up with a GAT, we're going to put alanine in. We're going to make alanine instead. And that's why that protein may have a different structure. Okay, so the alanine may not work as well as the proline for that particular amino acid, so it won't confer the right amount of function or integrity if it's a muscle or tissue, but if it's an enzyme, it may not be able to express itself properly. So this is how it works. In other words, you get a mismatch, which means we get a different amino acid in the protein structures. Now here is the code. So when we have an A into a C at the top there, which is very popular, A into C, it encodes for methylcobalamin. Okay, and what does methylcobalamin? It's involved with methylation or recycling of methyl groups. Those enzymes are found naturally in the pips of the orange pepper. Okay, and sometimes there's a virus or an infection that can cause this polymorphism, and the one that does this is rubella or German measles. It does not mean that everybody who weakens to A into C has had or got rubella. But rubella, this is how the virus can damage it. But the damage could be done from a toxic metal, it could be damaged with radiation. Doesn't mean, I've just given you examples of the infection. Okay. Now, if I could have my patient back. Okay. Now, I need, Jane, could I have one of the new boxes? Because I think that's got the markers in. That one, yeah. Yeah, how many you got?
One, about half a dozen.
Okay, we'll have 1, 2, 3. So we'll just do, while I'm doing this and demonstrating it, we'll do the zinc taste solution. So, one for you. Okay, one for you and one for you. So what you do with the zinc taste solution is you fill the pipette up with zinc, you open your mouth, lift your tongue off the roof, stick it out, that's it, squirt it in, don't suck the tube, because it's going to the next person, all right? Keep it in for 10 seconds. You can swill it round, and then hand it on to the next person. About 10 seconds, and then swallow it. And if you go yuck, and it's like a driest rhubarb you can imagine, that's good, okay? If it's only a mild yuck or no taste at all, that means you're zinc deficient, okay? So hand it on, and when at the end, we'll see how many people are zinc deficient. Okay, so if you pop yourself down. So what we're going to do is we're going to use the kit. No, this is the wrong kit. Jane, it's the new one, the one that's got the DNA, the markers. I got that one. So what we're going to do, we potentially got a candidate. The time when you do this will become a bit more obvious. I'm just doing it now, so it's not necessarily in this sequence. When would I decide to check for a polymorphism? The answer is whenever a coenzyme shows up. So if I'm going through you and analysing when you need a coenzyme, and pyridoxal 5-phosphate shows up, then I know there's a polymorphism. Okay? So what I'm going to do is I'm going to, in no particular order, I'm going to put adenine on first. Pull. Okay. Now, every colour has a complement to it. In other words, if you're blue, the complement to blue would be everything that's not blue. In other words, it would be green and red. So they all have complements. So the complementary colour to that violet end of the spectrum is a yellowy green. Right? Does everybody know what a complementary colour is? So there's 3 prime colours in optics, red, green and blue. Okay? So if you take red out of the spectrum, you're left with green and blue, which makes turquoise. So in other words, red and turquoise gives you the whole complete match. So it's called complementary colour. So when you put the turquoise on, you don't see the red. Okay, it absorbs it. So with a violet blue person, the opposite to blue will be red and green, which is yellow in optics. It makes yellow. But because it's more towards the violet end of the spectrum, we move it round so it's more the yellow end of green. So that's the complementary colour. So this is what we call a good colour. So this will absorb the blue. So what this will do is make you at your very best. So there's no blue light going in now. So what I'm going to do is when I test for toxins or adverse substances, I like to put the complementary colour on. Okay? So we'll now test with A, or adenine. They're nice and strong. And then we got C, or cytosine. And then we've got G, or guanosine. Good, so all strong so far. And then we've got T, thymine, and then we got finally U, or uracil. Save the best one to last. Okay, now we don't know, don't jump to any conclusions, okay? So we're weakening to T. Okay, so what do I do now? I put on the other ones one at a time to see which one negates. So we put on T. So this is not a U into T. So we now put on G. Put on C. So we got a U into C. And we've got an A. No. So we've got a U into C. So what you do now is you go back to your chart. Gary, if you could go back onto that one. So you go U into C is a combination of folic acid and vitamin B3, which is tetrahydrofolate, U into C. So this is her polymorphism. So she needs an active form of folic acid, which is called tetrahydrofolate. And if you follow this along, what you find is a 1-carbon transfer substance. It's rich in raspberries, particularly in the seeds of raspberries. And, look what the disease might be. So, does not mean she's got that disease or that she's inherited it. Oh, it's not. Is that one? Adenine. Oh, I must have jumped that one. Ah, dear. Okay, so adenovirus. Now, adenovirus is one that does the rounds in the autumn and the spring. Okay? If it catches you in October, when the climate changes, okay? Now, this October there was nothing at all really. And Christmas time, really very little problems at all. January, a little bit here and there. Wham, February. You know, every other person is coughing, aren't they? Okay? Was that adeno? No, not yet. It hasn't come round yet. Okay, it's like the comet, hasn't come round yet. That was influenza. And what an influenza outbreak. Okay? Some people it laid up, didn't they? 3 days, 4 days in bed. They were the lucky ones. They got better. Okay? I got the shakes a little bit, you know, that hot and cold feeling, and no sore throat, but the cough. And the cough lasted 3 weeks or more. You know, other people only had the cough. Other people got sore throats. Some got earaches, but it's all influenza virus. And we found eventually the answer to what it was that the body needed. And it's not a medicine that people need. They think, I want an antibiotic. Antibiotics are useless for viruses. What about echinacea? Didn't touch it. None of the usual things touch it because we're tackling this the wrong way. We don't need to kill the virus. Okay? We don't need to kill the bacteria there. We need to up our immune system, okay, to do it. What was happening is we'd had lousy summer after summer after summer. We're all tired. We've got no energy in there because we haven't had enough light. We haven't had enough vitamin D in our systems. We're knackered out. And if we're knackered out, our immune system's knackered out. And you need to up-regulate your immune system, okay? And that means up-regulating you to give you the right things so that you can produce your ATP. And once you found that combination, which we'll talk about later on, then things start to heal up. And so in the more advanced sort of stage of a malignancy, you need the same thing. You need to upregulate the person's immune system. And this is how we do it. So in this case, she has got a polymorphism. So we're going to have to take folic acid into consideration. Now this is an active coenzyme, and to do this we need a combination. We need folic acid, but we also need some NADH, or the active form of vitamin B3. So here what we would do is we'd mix folic acid. You put on folic acid and vitamin B3 or niacin. Okay? Now to activate NAD and to activate you need zinc. So you may need all three. So if you want to use and make tetrahydrofolate, because you can't buy it, it's totally unstable. You have to make it yourself. So you would make it by putting on the folic acid vial, okay, the niacin, and probably the zinc in here. The body can make most of these, but there are some which it can't very easily. So pyridoxal 5-phosphate it can't make very easily. Adenosylmethylcobalamin. These are the common coenzymes which very often are true inherited polymorphisms that you have to take as an extra supplement as such. The body is unable to convert enough of it for that particular person. Most of the other ones, in our experience, they can make, providing you give them the building blocks to do it. Most of the folates are dead easy to make. You need zinc is the key to it. So when you make methanol or folinic acid, you simply use folic acid and zinc. A combination of folic acid and zinc will make most of the active forms of folic acid. You don't need hideously expensive methanol. The body can make it. And all you've got to do is test by putting the 2 or 3 substances on the person, and bang, it works very well. So in your case, you do have a polymorphism. We've got to keep an eye, and we would expect the folates to be showing up. Okay? So that's one component of her. Although we've taken it in isolation, you will need some folic acid, that's for sure. Okay? So we've got a lot to do with you. Okay. So... So that's how you check for a polymorphism. Okay, yeah, at the moment. Okay, so adenovirus. So adenovirus, if a person shows to a U into a C, there could be that the person's had an adenovirus and it's had an effect within the DNA structure. Because it's a virus, it is a coding of DNA or RNA, and it could get in and change the program. It does not mean that she's got adenovirus, or it doesn't mean that she's had it. But she could have, okay? Equally, remember, it's not just viruses, it's toxic metals, radiations, chemicals that do this, okay? But we do know for sure that with this one, if there's a virus involved, it's the adenovirus. So you can see from here, the type of viruses and some bacteria which contain viruses are the things that can cause the polymorphism. But remember, it's not round the other way. It doesn't mean everybody with a polymorphism has got syphilis or gonorrhea or had it in the family even. Okay. So assessing the correct optimal nutrient is you go back to the chart there, and I assessed there it was a U into a C, and looked on my chart there and said, that's tetrahydrofolate, and then what I did is the weakening nucleotide on, so we left the U on there, and we challenged them with the appropriate coenzyme. You cross-therapy localize now to the 3 major points. Now I use 3 points in my work that I discovered were very, very useful for making sure something is effective for a patient. So if they strengthen to it, it means it's working. But is it really working everywhere, especially in the left brain and the right brain? Get them to therapy localized conception vessel 22, which is on the top of the manubrial sternal. This is the point we use for the corpus callosum. So if the person goes strong, she went strong to the folate, touched there and went back into weakness, it would mean it's no good, it's not effective. Okay? So that was the first one, the corpus callosum. Does it cross over to both places? GV20, which is traditionally the point we use for the hypothalamus, the regulator. And again, if they stay strong, it's good. If they go weak, forget it. And the other one is putting the tongue on the tip of the mouth, get on the roof of the mouth, go back to the soft palate and come forward about 1 centimetre. And you should be right on the cruciate suture then. And there's like a little bony lump there, torus sometimes. Touch there, and that's for the pituitary gland. So what you're doing is you're just testing. Now, I know other people who do different schools of kinesiology like to use finger modes, sometimes priority ones, and sometimes people use hands out there and so on. Do whatever you want, but make sure you've got something which fulfills the criteria. So in other words, let's say for pyridoxal 5-phosphate, a person has a genetic predisposition of polymorphism of pyridoxal 5. They may just need more B6 and magnesium, because magnesium is the cofactor to get B6 activated to pyridoxal 5. If they only strengthen to pyridoxal 5-phosphate, it'll hold on all three of those. If they can't convert and you put pyridoxine on with magnesium and they go strong, they will probably weaken to one of those if they can't make it. Okay? So this helps you to determine whether the person needs the activated vitamin in their diet or as a supplement, or the inert one and the cofactors to get it to work. Okay? Does that make sense? So, okay. A little bit hard to understand, I know, but there are times when people can't activate the vitamins in their way to make them into coenzymes. And there are times when they can. All they need is the vitamin and the cofactors to do it. So here, and you have a very small, this again is a chart, it's a laminate that we do, and I think it's on the website to download. In addition, this just shows you how you activate each vitamin into a coenzyme. So we looked at folic acid, which is the next one there. We looked at the activation just now of the folates. Okay, so this chart shows you how we activate pyridoxine into pyridoxal 5-phosphate, how you activate B5 into CoA, and so on. So here we see thiamine going on to thiamine pyrophosphate and thiamine triphosphate. So they're all in there, all the information. All the hard work is done for you. Okay? You just got to basically copy the rules. Okay? Now, sometimes there's individual variation, but the answer usually is magnesium or zinc. There we go, I've given it away. Okay? That's the most important deficiencies that people show to. Okay? Why? Because of intensive farming. Now, I teach in America, I teach in Australia, Russia, and they're all the same. Everybody in Australia is magnesium or zinc deficient, Russia, America, because of intensive farming, because we drained all these minerals out. And when they put back the phosphates and the nitrates, but they don't put back the zinc, do they, and the magnesium. So very gradually the soil is getting more and more depleted, and that's why more and more people then can't activate the other vitamins into their coenzymes. So they show up as folic acid deficient, they show up as vitamin B6 or pyridoxal 5-phosphate deficient, because we are deficient. Okay? But, you know, most of you are sensible and can pick these things up and start to rectify and take the right sort of supplements to help, or eat the right foods with it. So I am 2.85 billion nucleotides of DNA, and all this DNA encodes only about 20 to 25,000 protein coding genes. And this was interesting because it was assumed that there were going to be at least 150 codings, 150,000 codings from the Human Genome Program. And so it was quite a surprise when only 20 to 25,000 came up as far as this is concerned, which means that each gene can encode for a different number of amino acids. Okay, so we could produce different types of protein from the same gene. Okay? Now, an amino acid, or the gene, depends upon which part of the gene is expressed or exposed. So if you were to read, this arm here was the gene, okay, and it's made up of a series of nucleotides. So we've got TTT, CTP, and so on, all different codes for different amino acids, which ultimately could be hundreds or thousands of amino acids long to make a protein. Now, if only that bit is encoded and the rest is covered in a protein sleeve, as it's called, you can only see my hand. Okay? Therefore, that can only be read. So the DNA sequence in that hand is for what's going to be read. So that will make a particular protein. Now, if I do that, I make a different protein because I'm adding other amino acids. And if I then hide that bit and only expose that bit, I get another. So each gene can, in fact, encode for multiple proteins. Many, many more than was ever imagined, okay? Because the Human Genome Program wanted one protein per gene, okay? Why? For the health of the nation of the world. No, for corporate business, because they wanted to patent every single drug. That would be one drug for everyone. So never be fooled that the Human Genome Program was for the benefit of mankind. It wasn't. So it depends upon which part is opened. Now, how do you open the gene to get it to express? What pulls the protein sleeve away are little cords mechanically which are connected to the cell membranes. Now, the cell membranes have receptor sites, and the receptor sites are made of proteins. And when a ligand or substance binds to the receptor site, it changes the conformation. Now, proteins are made of positive, negative, and neutral amino acids. In other words, they have charges, which gives them a shape. So they're a bit like a drain pipe. So you have a bit coming out here, then you have a right angle bend, and then another one. Okay? Now, if you add a ligand or substance which changes the charge, the protein changes its shape. And that literally mechanical side of changing the shape pulls the little strings which operate the gene. Okay? And they operate the protein. So therefore, you could have a substance lock onto the receptor site on the nuclear membrane, and it'll do this. And another substance on there will do this. And another one will keep it right down there. So toxic substances tend to shut things down. And good substances like nutrients tend to expose it and open it up. But what we don't want is to open up that oncogene, isn't it? We want to open up our tumour suppressor, but we don't want to open up the bad ones. So we want to keep the sleeve hidden on those. So what keeps that sleeve nice and tight around there are proteins called histones. So we want to lock that cuff right down in there, so we don't want to expose the nasty bit further up there. And what holds that down is a methyl group. So if you're deficient in methyl groups, this becomes a little looser and might start exposing itself. Okay? You got to 40, 45, and you're deficient in methyl donors, like the B12s and the folates and the B6s, etc., and you're starting to expose bits which you don't want to expose. Okay? So to keep the caps on, or the sleeve tight there, we need methylation. And this is really where methylation comes in. So epigenetics shows that there is more to inheritance than genes alone. So in other words, these are epigenetics. Epi means above. Genetics means things above the gene. So things that can modify the gene's exposure for good or bad. And of course, the big epigenetic phenomena are the toxins, are the deficiencies, and the emotions, of course. So there's more to inheritance than genes alone, opening the doorway for environmental and other lifetime effects to be transmitted. And this was proposed by John Baptista Lamarck in the late 1700s, early 1800s, long before we really knew about genes and genetics. So it's really interesting that a lot of this is not new. So epigenetics are mechanisms that lie outside of the DNA sequence. One of the initial discoveries was the effect of DNA methylation upon gene expression, and then modifications of the nucleosomal histones. This DNA methylation usually is associated with the cytosine part by methylating cytosine. So this is what I've been talking about to show you. Okay? So here you see the DNA ladder here, which we're familiar with, and here you see the proteins, the histone proteins. They have little tails, and it's on those tails that they can be pulled and have tags put on. So this loosens the DNA, the expression of it, or tightens it, as the case may be. These pictures, if you put in epigenetics on the internet for pictures, these are all on there. So there's nothing patented about this. But you can see these lovely histone tails and how they can be loosened if you're deficient in methyl factors. So each histone has a loose end or tail, which certain chemicals can attach, which alter how tightly the DNA is coiled. So long as the DNA remains tightly coiled, the gene does not activate, but to activate it, if the DNA must be partially unwound. The gene needs outside instructions from acetyl or methyl groups. Sometimes acetyl groups are added to the tail near the gene, causing the histone to loosen its grip. So in this case, acetyl groups loosen it and allow it to express, and that might be good in a tumour suppressor gene. We want it to express, whereas a deficiency of the methyl group will allow bad ones. So removing the acetyl group causes histones to tighten their grip on the DNA, therefore stopping the expression. Put simply, adding a methyl group switches the gene off, and removing a methyl group switches it back on.